So this will be the first topic here will be discussing the yeast model we generated. So in yeast, PMM2 goes by a different name, Sec53. So I may use those terms interchangeably, but we're obviously referring to the same thing. And just a little bit of context here, we actually worked with a contractor that focuses on yeast work called Next Interactions. We met them through QB3. Actually, the former operations manager of QB3, Rich Yu, was a yeast geneticist in his past life, and they started a company, essentially a contracting company to build yeast strains. So we actually use them because we're still building our own yeast team internally at Perlara. So we use them to contract out the creation and validation of a set of PMM2 yeast strains. And that's what I'll describe here in this first part of the talk. So just a little bit of background. So I know everyone here is not a yeast geneticist, and so there will be a lot of nomenclature and other descriptions here that are maybe foreign to you. So I'll try to translate and try to keep things as non-technical, non-jargon as possible. But I also hope that with these calls, we've got a range of expertise and background here. So I'm hoping that we try to eventually equalize the playing field. Can I make everybody a PhD? But I think ultimately we do want to be using specific terms when they are appropriate. So ultimately everyone will be fluent in genetics at some point and other aspects of drug discovery. In any case, this is just some of the background about how we decided to create a bunch of mutants in yeast. We identified three different mutations, three different point mutations that we were interested in modeling initially. I mean, we can obviously model whatever mutations we like, essentially. There are some constraints there, but these are the three mutations that we initially started with. R148H, or I guess here, we're actually showing you here two sets of numbering schemes. So the parentheses are the human, refer to the human PMM2 gene position. So for example, R141H is R148 in yeast. So as long as you don't get tripped up on that, we pick three mutations. So I'll just use the human numbering system. So the V231M, the R141H, and the F119L. And we were inspired by the mouse model, the R141H F119L compound heterozygote that was made by Agios, and the kind of thinking here was to try to create these simpler models that are aligned genetically to the mouse model, because ultimately to really translate this beyond the preclinical phase, we need to be able to establish some kind of efficacy in a mouse model. And there's a bit of trickery we had to use to make this work in yeast, because the PMM2 gene is essential for yeast growth. Without PMM2 or Sec53, yeasts just don't even grow at all. So it's a quote unquote essential gene. So we had to do some tricks to be able to express these mutant forms of PMM2 in the yeast while being able to carefully turn on or turn off the normal copy of PMM2 or Sec53. Because if we just delete the PMM2 in the yeast, they just will not grow. And so we can't do any drug discovery with an inviable strain. And in the next slides, I won't try to go into too much detail here, but we'll at least give you a sense of how we do this kind of strain construction. And so the bottom line is the question that we pose at the bottom. And again, this is using the more technical genetics language, but the idea is if we make if we can make a knockout of PMM2 in the yeast, and we put in the place of that missing PMM2, we put in a mutant form of PMM2. The question is, does that mutant form of PMM2 have enough function that the yeast can survive? Because we've deprived them of their normal PMM2. And so that's really the setup of this system, is we're able to introduce both a normal copy, a normal version of the PMM2 gene in yeast, and then a mutant form. And then we can turn off the normal form of the gene and then ask if the yeast still survive, if they have to rely on the mutant form. Is that kind of clear to everybody? Hopefully it will become more clear as we go. But essentially, the nice thing about yeast is that it's really a screen where the readout is, are they alive or not? And so it's the simplest kind of screen you can do, and we think it's actually a very powerful screen and a very fast screen to run. So this was just some data we pulled to try to get a sense of the different residual activities of these different PMM2 mutants. And so, we knew that the R141H was sort of the most inactive, was the most severe mutation, and we knew the other mutations would have some residual activity, and that they probably had some kind of misfolding or protein stability problem, and that was the nature of the loss of function, or the nature of the hypomorph mutation. So we didn't want to model all the mutations up front. We wanted to first pick a few representatives and to understand, are we actually able to show that mutant forms of PMM2 are if they're sufficient for life in a yeast cell. So we generated a whole list of strains on the left. And the thinking here was that we wanted to create the 3 different mutants, the R141H, the F119L, and the V231. Again, numbering system here reflects the position in the yeast genome or in the yeast protein. So don't get tripped up on those numbers. And so we're testing these 3 different mutants plus a control. The WT stands for wild type. That's the normal copy. And here I'm referring to Sec53. So this is all using the yeast language of describing the PMM2 gene. So we've got so essentially have two controls, a wild type Sec53, and then just an empty plasmid, just GFP. So there should be no rescue from that plasmid at all. And then we've got the 3 different point mutants. And so what we've done here in these 4 batches of 5 is that we've created different strength promoters to drive the expression of these Sec53 variant genes. So we use the quote unquote endogenous promoter. In other words, we use the promoter that's normally in the genome in the location of Sec53 that regulates how much of that protein is generated ultimately in the cell. Well, first, how much of the transcript is created, and then ultimately how much protein is in the cell. So we use a number of different strengths promoter. We use the endogenous promoter, the native promoter, and then we use kind of a weak, medium, strong regimen. And the names of those promoters are all referred to by that P and then those initials. So PACT1 or PREV1. Again, don't worry about those. Don't memorize those names. It's just important to know that we've got a range of strengths here, from strong to weak, and then of course the endogenous or the normal promoter for Sec53. And on the right, we're kind of showing a very simple schematic of a yeast cell. And the concept here is to say that, again, we have to, because Sec53 is an essential gene in yeast, you can't just break it and expect the yeast to survive. So you have to, if you want to test a variant of Sec53 in the yeast and ask if it can support growth, you have to actually have a normal copy of Sec53 and then the mutant copy in the same yeast cell. Now, the mutant copy we would put and integrate into the genome. And that's why we refer to that, we sort of show an orange bar there to kind of represent that this is an actual integration into the genome. And then the normal copy of Sec53 is kept on a plasmid. That's shown here by this orange circle structure. What's nice about putting the normal copy or the wild type copy of Sec53 on this plasmid is that we can control the expression of the wild type Sec53 very, very reliably. And effectively, we can turn off the wild type Sec53 so that the only Sec53 function in the cell would come from the genome encoded copy that is the mutant form, right? Either the R141H or whatever it was. And so we have some tricks that we can use to turn off the normal copy of Sec53, and that's what I'll get into. So the idea here, again, I don't want to turn you guys into yeast genetics experts, but all you need to really know is that we have a way to turn off the Sec53 wild type faucet, so to speak. And that's by adding something to the media. It's a compound called 5-FOA, and it's a nice genetic trick. I won't go through all the details, but it allows you to essentially turn off the spigot for the wild type Sec53, so that the only Sec53 that's being produced in these cells would be the mutant copy, the one that's on the genome. And so the idea would be is that we create these mutant strains. They've got the wild type copy and the mutant copy of Sec53 inside each cell. And then we can, with an environmental condition, by adding this drug, 5-FOA, we can turn off the wild type Sec53. And then the question is, do the yeast grow? If they don't grow at all, it suggests that, well, the mutation that we put into that Sec53 that's still in the cell is not allowing the protein to function enough to support life. But if you do see some growth, it tells you that, hey, the only form of Sec53 in this cell is this mutant form, and there's enough activity left to support life. And so that's the question. And we expect, again, given the severity of the mutations, and given the fact that we can control how much of the mutant Sec53 we express, we could kind of test a whole bunch of conditions to understand, are there any parameters under which we see a mutant Sec53 support yeast cell growth when there's no normal Sec53 around? Okay, so just, again, one brief slide on the F5OA. It's science for 5-fluoroaromatic acid. Again, it's just a way for, it's a nice trick that yeast geneticists figure out as a way to turn off one copy of a gene while leaving another copy on. That's all you really have to know. So the design of the experiment was to grow up some yeast. We first grow them on solid plates, solid petri dishes. We grow the yeast up to a big, rich suspension. So there's many tens of millions of cells. And then we dilute those cells. We add that 5-FOA. That turns off the normal wild type Sec53 and allows for the mutant Sec53 to be expressed. And then we do different dilutions of the yeast. So in other words, just changing the numbers of the yeast from more to less. We do an incubation, and then we just simply ask at the end of 4, 18, 27, 48, and 72-hour periods, we ask how much yeast grew. And next slide is just showing you kind of the design of the plate. We included, obviously, the replicates required to be able to believe this kind of data. And so the nice thing is that with all these mutants and all the dilutions and all the conditions, we can fit essentially on just one plate, 196-well plate in this case. Although we could do this, and we will do this screen in 384-well format, but this was really just for the model development stage. Here's a control. So I said before that we were able to not only test different Sec53 mutants, but also test different promoter strengths driving those mutant Sec53 proteins. And so this is just data showing you that indeed we do have promoters that have different strengths. And all you should really be paying attention to here are the plots on the left, the cell count versus the fluorescence. So in green, in the table on the right, you see in green the PREV. This represents sort of the low or the weak promoter. Okay, and so it gives you a GFP or an intensity measurement, in this case of 18 arbitrary units. The negative control is black. That's sort of like your true baseline. So that's essentially no activity from the gene. So Rev1 represents that weak first step, and then you kind of do a step up in blue. That's the P-Sec53. So that actually is the endogenous Sec53 promoter, and that's 80 arbitrary units. Then we have in purple, that represents our sort of medium. So it turns out that the Sec53 endogenous promoter is essentially equivalent to what looks like a kind of medium strength promoter if you just are thinking about the strengths of promoters. And then the highest strength promoter we have was this PTF1. And you can see here that produces an intensity 10 times higher than the normal endogenous Sec53 promoter. So we're really testing a wide range of protein levels, of Sec53 protein levels, and of course we're testing the different mutants across these promoter panels. So here's a summary of all the data, and I'll break it up into chunks so you can actually interpret it intelligibly. But essentially what happened was all the controls all checked out as we kind of expected. And there was one contamination, which was can totally happen here, and I'll explain that. But that was just sort of an artifact. But other than that, all the other controls and all the other conditions worked really, really well. And so we're really, really confident that we've created a set of PMM2/Sec53 mutants in yeast that are going to be viable and very amenable to drug screening. So let's just break up the strains. Again, there's 5 different strains per promoter condition, and we're going to be looking at different numbers of yeast. So we tested, different concentrations of yeast cells, essentially, 10,000 yeast cells per well, 1,000 yeast cells per well, 100,000 yeast cells per well, et cetera. And so here I'll just break up the data and show you what actually is happening. So let's walk through the upper right panel first, so the endogenous plot. So what you're seeing here, again, you can't really see most of the strains here because they're not viable. So what's growing here in the endogenous plot is the blue line, and that represents wild type Sec53 under its normal promoter. So this is sort of like saying we knocked out the Sec53 function with this 5-FOA trick, and so there's no Sec53 around the cell. And so the blue line just says we're going to put it right back in. So this is your real positive control. If this doesn't work, then something very screwy is happening. So that's really assuring to see that the Sec53, the blue line, is always sort of reaches the highest level. So what we're looking here of the scale here is essentially a scale that corresponds to how much yeast are growing. So in these kinds of experiments, the plot usually levels out at 1. So that means maximal growth. And then obviously anything below that would be intermediate to no growth at all. So it's reassuring to see that as you increase the strength of the promoter, you always see that the blue Sec53 is sort of the winner at all times. And that makes sort of total sense. Now let's look for a second here at the other control. So the blue line represents the wild type Sec53. The red line represents an empty vector. So you're not replacing with anything. So that should be your negative control. So in all the plots except the strong plot, you can see that the red line is essentially invisible because it's completely flat. And it doesn't matter whether you have Well, here's where there's an artifact. So in the lower right quadrant, so the strong plot, you can see that the red line is visible. But I can assure you, we looked at that in the wells of those plate, and we saw that that growth, quote unquote, is actually coming from bacteria, not from yeast. So we could actually go back to the data and probably just artificially put that to 0, but we just showed you the real raw data because that's what we like to do. But we can explain that even though it looks like, wait a minute, why is the negative control up in the strong condition? That's actually bacterial growth. So that line should actually be down, just like it is in every other condition, whether it's endogenous, medium, or weak. So that is an artifact. And so in the next couple slides, you will see that the red line is up in the strong condition. But be rest assured that was just a fluke. The negative control was otherwise working as expected. Now, the interesting part comes when we ask, okay, we know the positive control grows. That's great. We know the negative control is not growing. That's great. Now, what about the mutants we made? Remember, we made 3 mutants of various strengths or different severity of loss of function, with the R148H being the most severe. And does that actually what we find? So we would predict that you should be able to see growth from, say, the F126L and the V238M at some condition, either some promoter strength or after some amount of time, you should expect to see some growth. But the green line, or the R148H mutation, the worst mutation, the most severe mutation, we would expect that one to be the least likely to grow, right? Because that should be most like a null. That should be most like effectively the red line. You're putting back the CYP3, but it doesn't work at all. And what's interesting is that we can see that in the medium plot. So let's focus right now on the medium plot, so the upper right plot. And you can see, of course, the Sec53, we've got our control condition. This is now looking at the medium strength promoter. And you can see here that the lighter blue line and the purple line are showing a kind of intermediate result compared to the blue and then the red, which would be the negative control. So it's almost like just exactly in the middle. And what that tells you is that the F126L and the V238M mutations are not so severe that they're a null. There must still be some Sec53 function left because it's supporting growth. Now, it doesn't support as much growth as the wild type Sec53, because what's interesting is that there are 2 features. One, the F126 and the V238 plots are shifted to the right compared to the blue line. So that means it takes longer for them to grow, right? So at 72 hours, the V238M is at 0.2 growth, whereas the wild type Sec53 is at 0.8. So you can see that's that window of difference here. But ultimately, the F126 and the V238 do catch up to some extent, but they don't get all the way to 1. They only grow to about half or 60% of the total sort of what we call the yield. The total number of cells is lower. So that's fine. I think the result we were looking for was exactly something like this. Because if you saw no growth at all, then these yeast are too sick potentially to be rescued by any intervention. But if you get this kind of Goldilocks result where it's growing more slowly than the positive control, and it doesn't get the same total number of cells, but it is actually viable compared to the R148H and the negative control, that was to us kind of the desired result, and that's what we found. And essentially the same pattern repeats again and again, but looking at the different dilutions of yeast cells. So in the 10 to the minus 2, that means we diluted the original stock of yeast 100 times, so 1 to 100. Then in this condition, 10 to the minus 3, we diluted the original stock 1 to 1,000, 1,000-fold. And then in 10 to the minus 4, we did it 10,000-fold. But essentially, again, you see the same pattern emerging again. So if you look again, and it's the same position of the promoters, right? So the endogenous is in the upper left, and then medium on the upper right, and then weak lower left, and then strong lower right. So again, in the lower right, the red graph, the red curve, the PTEF GFP, that should be the net control. Again, that's the bacterial growth, that's the artifact. So if you can just imagine that line being down, then you can interpret the rest of the results. So in the endogenous promoter, all you see is the Sec53. In the medium promoter, what you see is that actually it looks like there may even be a separation between the F126L and the V238M, suggesting that maybe the F126L suggesting that the V238M might, in this case, in this yeast context, is potentially a more severe mutation relative to the F126L. And again, the same essential pattern emerges at the 10 to the minus 4 dilution. But what's interesting here in the 10 to the minus 4 is that we found that in only looking at the case now in the lower right plot, in the strong promoter, again, if you ignore the red line, what's interesting is that if you actually have P53, excuse me, if you have Sec53 being driven by the strong promoter, so that blue line, it's actually doing worse than the two mutants. So that's interesting. That we didn't expect to see that result. What that means is that when you overexpress the wild type Sec53, so you just have the same Sec53 as normal, but you just have way more of it, that actually is, that can inhibit cells, that can inhibit cell growth. And so we can talk about how that could be significant. At a minimum, it serves as a good control condition because we can include the wild type Sec53 with this PTEF, with the strongest promoter. We can include that when we do screens, because we would expect that the reason why those strains are not growing well is not the same as the reason why the Sec53 mutants are not going well. The Sec53 mutants have a loss of function, but the Sec53 overexpressors, that's wild type, again, the enzyme still has the same activity. It's just you've got a lot more of the protein around. Why has that become toxic? You know, that's interesting. Why? I mean, it's not the first time that overexpression induces toxicity in yeast cells, but it was interesting to be able to observe that here. So that's sort of that's the extent of sort of the yeast data. So kind of where we go from here is that we can pick conditions, essentially time points, that we would want to build a screen around. And also build and also factor in how much yeast we need to add, because if you change the number of yeast you add, it changes sort of the timing when you want to observe growth optimally. So if we kind of go back to this condition, so 10 to the minus 3, so you grow up a whole bunch of yeast and you dilute that stock 1,000 times, 1,000-fold, excuse me, and then so essentially you're adding maybe 10,000 cells to the well, and you let those grow over a 72-hour period. What we would expect is that if we included the R141H as a control, that's a negative. That should never, we're assuming that that's not going to be rescuable because that mutation is simply too severe. And then if we include the F126L and the V238M, as well as and if we do all of those three with the actin promoter, which is the medium strength promoter, we think we would get this nice this nice time course. So that, for example, if you looked after 2 days, so let's say we worked with the F126 and the V238 strains, and we said, let's see the population at day 1 with 10 to the minus 3, 1,000-fold dilution, and then let's look at after 24 hours. And if we compare that to the Sec53 wild type control, you're already seeing growth at 27 hours with the Sec53 wild type, but you're seeing really no growth at all with the mutants. And so if you essentially did a time course and you measured the plates every day, starting after the first day, so at 24 hours, at 48 hours, and then again at last time at 72 hours, what would happen is that if you look at the 48-hour time mark, both the F126L and the V238M under the actin promoter have growth of around 0.4. So what that means is that there's still room for improvement. And so we could ask, would a small molecule or a drug at 48 hours cause the F216 to actually be closer to the wild type control at that time point versus waiting another day to catch up? So that would be kind of the rationale of the screen is, can we find molecules that allow for the F126L and the V238M to grow at closer to the same rate as the wild type Sec53? And then I said we could also include using the Sec53 and the TFEF, the PTEF, the most strong promoter, the one that has 10 times more activity than the normal Sec53 promoter. We could throw that in as a control because we expect that to have some growth defects as well. But that growth defect is due to the toxicity of too much PMM2, not due to the toxicity of too little PMM2 function. So we chose to make 3 different, make PMM2 models in 3 different species, knowing that the yeast would kind of take the least amount of time and get us to some data faster. And then the worms would sort of take the next amount of time, and the flies would kind of bring up the rear. So I'll give an update now on where we stand with the PMM2 worm strain construction. So this was sort of just text that we pulled out from the original PerlQuest research plan proposal. And as I already kind of described, we're looking across these 3 invertebrate species. And the goal here is to create a panel of mutations. A null mutant, which we actually expect will probably be very, very sick, if even viable at all. We propose to make novel hypomorph mutations by introducing premature stops into the PMM2 gene, so that maybe the gene is only made, only half the gene is made or 3/4 of the gene is made, and then you make a premature stop. And then we also wanted to create 2 point mutations. We wanted to knock in the 2 point mutations that correspond to the mouse model. Now, again, we don't have to limit ourselves just to these mutations, but the thinking was, let's make this kind of initial panel first. So I'll describe here actually the results of generating an F119L knock-in mutant. That was the first mutant that we made on top of the null. I don't really need to go, I think, too much into the background here. Again, the rationale for picking F119L was that this is a kind of classic hypomorph. So you've got some residual enzymatic activity left, not zero. So the idea is that potentially you could find therapies that would potentiate the residual function of an F119L mutant. And presumably that would be because this mutation is causing a protein instability or a protein folding problem. So if we could just potentiate or chaperone the function of that protein, you could achieve enough, you could get enough PMM2 activity back that you would have viability. But we could also model other point mutations as well. But we wanted to start again with the mutations that were going to align us most closely with the mouse model. So in C. elegans, the PMM2 ortholog has this alphanumeric name, which tells you that it actually hasn't been studied. So there's not much to go on in the worm field. In contrast to yeast, where the Sec53 gene has been known for 30 years, in C. elegans, we're not building on that same kind of foundation. That said, there were alleles that we were able to acquire that were available, that were sort of strong loss of function alleles. And the challenge with those is that they have a larval lethality that's very early. So it's very difficult to work with a model where they're dying that early in development. So again, this is the actual sequence of the nature of this mutation. So one of the mutants that we were able to get through the C. elegans Genome Center, or essentially their stock center, had this large 500 base pair deletion. And so that was one mutant we were able to look at. And unfortunately, this allele is simply too strong. So just showing a plot of worm development. And so here's an image of some of these PMM2 mutants, and they arrest as larvae. And again, we think that that may simply be too severe of a phenotype to suppress, although we certainly can try to use that going forward as a control to see if we can get any amount of rescue. But the worms are quite sick, and the problem is we need to generate lots of homozygous animals. But this mutation is homozygous lethal. So you have to grow up the carriers. Only some percentage of their offspring are homozygotes. You have to isolate those physically. And it's usually hard to do when you've got a homozygous lethal mutation. It's hard to get enough animals to do any kind of study because they're just simply dying so soon. So this is what led us to think, let's really make a point mutant, especially the F119L, because we know that should have some residual function. And since we know a null mutation is really severe, we'd expect the F119L to be potentially either no larval lethality, or you would have less penetrant lethality, or you would have some kind of delay later in development, post-larval. So we are in the process still of getting CRISPR, a license to do CRISPR internally for our own strain generation. So in the interim, we've been working with partners that are already accredited and able to use CRISPR technology. And so one such partner is Nudra. We've used them successfully in the past to make our Niemann-Pick-C mutants and mutants for other projects. So we asked them to essentially dial in F119L into the nematodes. And again, the numbering is a little bit different between worms and humans. So F119L in humans is F125L in nematodes. And this is just to show you that the actual, amino acid F phenylalanine is actually conserved between humans and worms. When you compare the human PMM2 sequence and the worm PMM2 sequence, that position F119 is conserved. So that's also reassuring to see that in the worms, that amino acid, if it were to change, would probably also change a site that's not normally supposed to vary. Just a little bit about kind of the procedure here to generate a knock-in mutation. It's very easy with CRISPR to sort of generate null alleles or to generate premature stops and other kind of destructive alleles. It's a little bit trickier to generate point mutations, but we can use a procedure where we inject a marker. In this case, it's a dominant marker called DUMP-E10, which allows us to be able to identify individual animals where we've created a mutation. and then in the end isolate a heterozygote, which we could then propagate to be able to generate homozygotes and hopefully propagate the homozygotes, hopefully propagate the mutant actually as a homozygote, which would allow us to have just larger numbers of the animals to work with in screens. So in the worms, it's all based on a micro-injection, similar to how you would introduce CRISPR-Cas and guide RNAs into Drosophila larvae. It's a similar type of procedure and concept. And so I can get into the details of this, but suffice it to say, we have a process here to identify individual mutant clones. And then we did some PCR to validate that we actually did do these mutations. I can follow up with the scientists in the audience offline if you want to dig into this. And of course, we'll share all these slides after the presentation is over, so you can have them for future reference. But we did some QC here to confirm that we actually did make the mutation. And then, of course, we can go in directly by Sanger sequencing and look at the actual traces, and we can convince ourselves that we did introduce a point mutation. It's a little hard to see in this scheme here, but if you can look closely and you know how to interpret these, we actually did create the point mutation and successfully verify it. So the current status is we have got 2 clones of this mutant in the lab. The strains are actually homozygous viable, which is good, as I said. That allows us now to propagate them and have larger numbers to work with every time we grow up a population. We don't see any larval lethality like we see in the null, which is good. That's what we expected. We don't see any strong growth defects. We don't see any strong gross defects. And right now we're phenotyping them a little bit more carefully to measure exact developmental timing, to measure the percentage of animals that reach every developmental milestone, and then to look at other behaviors, potentially swimming behaviors, both in liquid and in solid media, and other things like how fat are the animals. As the animals develop, they kind of have a fat lining you can see. There's a number of phenotypes that we are primed to look for and that we will be examining in this mutant. And we are confident that we will find some kind of developmental phenotype that is not so severe that we wouldn't be worried about being able to suppress it. So for the worm project, we were gonna, as soon as we understand a little bit more about how this mutant works, we will then decide whether we want to make the R148H or R141H mutation, because we already have a null in the lab. We know that that's really sick, and we know that the R141H is effectively a null. We didn't want to go ahead and make that mutant right away, but we will make that mutant after we've characterized this mutant. And of course, we would like to make other point mutations. Just like in the yeast, there is really not a limit to being able to put in exact mutations. One condition would be you'd want to make sure that the amino acid is the same between worm and fly, or between yeast and between worm and human and between yeast and human. And then for most of the mutations that are known that cause disease in humans, they're targeting amino acids that are actually conserved in the protein. It's not targeting random amino acids. So for most cases, if you take a pathogenic mutation in people, that mutation is affecting an amino acid that is conserved in these simpler animals. So yeah, we hope over the course of the next few weeks or so to be able to have fully completed the F125L phenotyping. And if, as we suspect we find a developmental phenotype, we already have all the assays built to measure developmental timing and developmental rate and developmental sort of reaching developmental milestones. So as soon as we confirm that any of those phenotypes exist in this mutant, we already have the assay built and ready to do a screen. And then the next question would be, what other point mutants would we want to make after we would do a pilot screen with this mutation? This is just telling you about the progress we've made with fly. So we had a little bit of delay in getting the fly mutant generated. The partner group that we're working with, Genetivision, the company, is actually based in Houston. And so they had some problems last month after the hurricane and some delays, but they're back on schedule. So we hope that within the next month or so, we actually have our first mutants for flies as well. So the flies will catch up. So this is just analogous to the approach we use in the worms. You've got to do, here's a scheme of how you would actually go ahead and make a null. And the null actually is the first step on the path to making a point mutant, at least in the fly. So in flies, what we like to do is start out by making a null that essentially replaces the whole open reading frame or the whole gene. And what we do is we want to replace the native PMM2 gene with a selectable marker, in this case, a red fluorescence marker, the DS red. So the idea here is that you know that you've made a mutant because these animals now are producing a red fluorescent protein. And so that's how you know that you've created a null. So that's step one in the process. So that was what was partly delayed at Genetivision because of the hurricane, and that now is back underway. So we should have this null mutant first pretty soon. And then once we have that null mutant, we can go ahead and then on top of that null, put back in mutant forms of PMM2 that have mutations that we desire. So again, we're initially thinking, let's try to align the genotypes of these simpler animals with the mouse model. So that would be, again, making the R141H and the F119L. In this case, again, the numbering system's a little different. It's R137H in flies and F115L. But the idea here is once you create this null, you can then use some genetic engineering tricks to then drop back in PMM2 with any mutation we would want. And so we'll start off with these two mutants. So it takes about a month to generate the null. That's in progress now. And then once we have the null, it takes about another month to generate the knock-ins. So we're pretty optimistic that we'll have fly mutants sort of ready to characterize, heading into the holiday season. And definitely by the end of the year, we should have them fully, sort of fully phenotyped. And if there is a phenotype that we suspect, which is a larval arrest or a larval small larva size, again, we've already developed the assay to do that kind of screen. So we'd be able to very quickly go from a validated phenotype to a pilot screen. So yeah, so those are all the updates for now, kind of taking it all in. The idea is that with yeast over the next month or so, we would perform our first pilot screen. And we're going to be focusing initially on a collection of known bioactive and FDA-approved compounds. It's called the Microsource Collection Library, 2,600 compounds. It's the first library we screen all of our models in. So we're working on 5 other programs right now, including in addition to PMM2, and all of those, half of them have already been put through the Microsource collection, and the other half will go through them as well. So yeah, so on the yeast side, we should have some potential hits from a Microsource screen in the next month or so. And then those hits would be immediately tested on the worm, and then once we have the fly mutants, on the flies. To see if there's any cross-hit validation. And then again, depending on the phenotypes that we see in the worm and the fly, we suspect that they're both going to be developmental phenotypes. We've built those assays already. So as soon as we've confirmed what the defect is, we would be ready to then move to a Microsource screen for both the worm and the fly.